Bismillahirrahmanirrahim. We start the June 22 paper 2-2. I believe this is a difficult paper. Some of you commented on that. So I'm trying to make my best to explain it in a better manner. As we all know, this paper is for 1 hour 45 minutes. And this is a theory paper, but there will not be any section B or C anymore in the new papers. So everybody should remember that. But the number of marks remains the same. It will be 80 marks for the whole paper. So no more section B and C, so no more essay questions. Diagram shows the human brain and the table lists the function of some parts of the human brain. Parts of the brain: A, memory, stage, storage and control of conscious behavior. B, control of body temperature. C, control of balance and coordination. D, control of unconscious activities such as breathing. Label on diagram the parts of the brain identified on the table using label lines and the letters A to D only. Now I've done that for you. Memory, storage, and control of conscious behavior, that's the cerebrum. Control of body temperature, hypothalamus. Balance and coordination as the cerebellum. And control of breathing and all is the medulla oblongata. Now this was 4 marks and the labeling got you your 4 marks. Then it says name one structure in the skin that receives impulses from part B of the brain to control body temperature. So erector hair muscle or sweat gland or the smooth muscle or the muscles in the wall of the blood vessels of the arteries or the arterioles which will vasoconstrict or vasodilate. So you don't say capillary. Capillary was a reject, so that means a large number of students must have written capillary. That's why it's been mentioned as a reject in the exam report and in the mark schemes. So name one structure, then outline how the action of the structure results in the control of body temperature. Now that was easy. Erector muscle contracts, raises the hair, traps the air, and reduces heat loss. If you're talking of the sweat gland, then it produces sweat and evaporates and cools the body down. If you're talking of the smooth muscle in the walls of the arteries, then it contracts, and that's called vasoconstriction. So less blood to the capillaries on the surface of the skin, and this will reduce heat loss. So it depends what you wrote in B1, and then of course you would do the same thing in B2. So if you talked of the hair erector muscle, then of course you would give me the points about that the muscles contract, raises the hair, and this hair traps the air or insulates the body, and this of course reduces the heat loss. So if you mentioned the sweat gland, then of course you would talk about how that works. So that of course the motor neuron to the sweat gland. You enter this room is very hot. So this information goes to the brain. The brain sends information through motor neurons to the sweat glands, and the sweat glands produce sweat. The sweat will evaporate and will cool the body down. So name one structure in the skin that receives impulses from part B of the brain to control body temperature. So it must be the motor neurons. Motor neurons supply muscles and glands. Always remember M motor, M muscle, and glands. So motor neurons supply muscles and glands. And the other third option was, you see, we have 3 options which you could have written. So the third option was smooth muscles in the wall of the arteries or the arterioles. Now what will happen there? The muscle will contract. So the artery muscle, when it contracts, it makes the lumen smaller. So vasoconstriction. So less blood will go to the capillaries in the skin surface. So less heat will be lost. So reduces heat loss. So this is how vasoconstriction occurs when you enter a room and that room is very cold. So this is a way to conserve the temperature of the body so that your temperature remains at 37 degrees Celsius and you do not become, your temperature does not drop as the environment temperature is lesser. Maybe the room is at 30 or 29 or 28 or 26. So your temperature will remain at 37. Now coming to question number 2, the diagram shows an apparatus used to study breathing. We have a revolving drum, we have a pen, there's an upper box containing air, there's a tube to the mouthpiece, then there's air to and from the person, then there's a substance M, and there's water. Describe what will happen to the upper box containing air as a person takes one breath in from the apparatus. So what is the upper box going to happen? The upper box is going to fall or is going to drop down. Or you can say drops. Because when a person, it says when a person takes one breath in, so air is going to be taking breath in. When it's going to take, when the pressure is going to decrease, this is going to go down. State the percentage of oxygen in the atmosphere here. It's for 20%. You could have said 20 or 21, that's fine. Then it says explain how the percentage of oxygen in the air in the upper box will change as a person breathes in and out through the apparatus several times. Yeah, it will decrease. Why? Because the oxygen is entering the alveoli and then entering the blood. So decreases. Why will it decrease? Because it's going into the blood. So first, how does it change? It said. So explain is how does it change? So it's going to decrease into the blood. Why? Because it's needed for aerobic respiration. You had to say aerobic respiration. You don't have to just say respiration. Aerobic respiration, because the oxygen, we're talking of the oxygen. Then it says substance M is present in the apparatus to absorb an excretory product from expired air. Name the excretory product. Carbon dioxide. Yeah, because oxygen is being taken in and carbon dioxide is being released. So that has to be absorbed so as to maintain the level. The diagram shows a chart recorded by the pen on the revolving drum. The chart was recorded for a period of 1 minute by a person at rest. So time in seconds, we've got a minute, and volume of air in dm cube. Explain how the action of one named muscle caused the part of the chart labeled N. Now, what is N? This is what you've got to figure out. Now, if you remember in the first part of the question, here, what did it say? Describe what happens to the upper box containing air as a person takes one breath in. We said it drops, so that means what is going to happen to the pen? If it drops, there's going to be a line which goes down. So the down line is an inhalation. So this is inhale. So if it is inhale, explain how the action of one named muscle causes the part of labeled N. So first of all, we've got to give it for inhalation. So in inhalation, what is going to happen? Diaphragm muscle and the external intercostal muscles contract. The diaphragm becomes flattened, ribs move up and out. So there's increase in volume and decrease in pressure of the thorax. And air is breathed in, air into the lungs, or we say inhale, and the upper box falls and the pen moves down. So this is what you have to gather from the first part of the question. So in order to get your 3 marks, you have to give me all those points. So how would we word this? Diaphragm muscle contracts, diaphragm moves down, increase in volume and decrease in pressure of the thorax. So this results in air being inhaled in and the upper box falls and the pen moves down. Upper box is part of the machine that we saw here, the upper box which is given here, upper box containing air. So the upper box falls and the pen moves down. And in part 2, a second chart was recorded for a period of 1 minute by the same person after vigorous exercise. State how an increased rate of breathing after exercise would be shown in this. Well, more waves, more peaks and troughs, or waves closer together, or an increased frequency of waves. Now we come on to the third question. A student carried out 2 investigations into the action of yeast. In the first investigation, the student dissolved 20.0 grams of glucose in 100 centimeter cube of water. In a beaker, the student then added 3.5 grams of yeast and used a balance to measure the loss in mass from the beaker over the next 24 hours. The results of the investigation are shown in the figure. Mass lost in gram and time in hours. So it is a maximum of, we used it for 24 hours. We've got the figures for 24 hours. Over next 24 hours. Now calculate the rate at which the mass was lost during the first 6 hours. And you've got the space for working and the units have been written for you. And how are you going to calculate this? 4.5 divided by 5 divided by 6. Why did we do that? We look at the end of 6 hours. This is what? Read this off. It's 4.5. So 4.5 divided by 6. Why? 6 hours. So this was 6 hours. So 4.5 divided by 6. And the answer would be 0.75. Explain why the mass was lost during this investigation. Why would you lose mass if you were not eating anything for 3, 4, 5 days? Well, what is happening is that the glucose in your body is being used up. That is being respired to release energy. That glucose has some mass, and that's being used up, and we are exhaling the carbon in the form of carbon dioxide. So the carbon which was part of the glucose molecule is now part of the carbon dioxide molecule which we are exhaling. So basically it was respiration, or you could have said fermentation as well, because we're talking of yeast. And then the next point was carbon dioxide. This carbon dioxide also was once part of the glucose molecule, so it had some mass. So carbon dioxide released or lost or escapes from the beaker. So the same glucose C6H12O6, now the C is being lost as CO2. So that is why there was a loss in mass. The student then repeated this first investigation, changing only the mass of yeast. Draw a line on the graph on page 6 to show the pattern of results you would expect the student to obtain when using 7.0 grams of yeast. 7.0 grams of yeast. Now you must have to remember is that here we have 7 grams of yeast, and here we had 3.5 grams of yeast. So more yeast means more carbon dioxide being produced, so more mass will be lost. Initial mass will be more, so more mass will be lost. There was 7 grams of yeast, more yeast in it. So the line of it is increased, is going to be the left of that, and will start at 0 and levels off at 8.5. Now you've got to understand why. At 4 hours, the loss was 3. Now the loss is 4. Why? Because there's more yeast. So there's more yeast, so there's more carbon dioxide being produced, more loss, so there'll be more mass lost per unit time, or you can say per hour. Now just trying to explain this to you, say in 3.5 grams of yeast, there were 1 million yeast cells. Now in 7 gram, there'll be 2 million yeast cells. So more yeast cells means more respiration, more carbon dioxide being lost, so more weight loss will it occur. So it's just, you've got to understand this is the mass lost. More cells respiring, more carbon dioxide, more glucose being respired to release carbon dioxide. In a second investigation, the student prepared 3 samples of bread dough. Each sample of dough had a volume of 50 cm cube and contained 1 gram of yeast, 25 cm cube of water, and 40 grams of flour. Starch molecules in the flour are used to provide the yeast with a source of glucose. Describe how the action of a named chemical produced by yeast cells makes glucose available from starch. Naturally, how is starch hydrolyzed, or how is starch digested? So we need the 2 enzymes amylase and maltase. Why? Because amylase converts the starch to maltose, and then we need the maltase to convert it to glucose. So we need 1 mark for that. And what does it do? It digests. Or you can use the word hydrolyzes, or you can even say breaks down. I don't like the word breaks down, it looks very silly, looks very unscientific. Hydrolyzes or breaks down the starch. So we have 2 marks, and you have 2 mark scheme points. So you could write either of these. You could write this point, or you could write this point. Each sample of dough was placed at the bottom of a 200 cm cube measuring cylinder. The 3 measuring cylinders were then placed in water baths at different temperatures for 60 minutes. The temperature chosen was 20, 40, and 80 degrees Celsius. After this time, the student measured the volume of dough in each measuring cylinder. The diagram shows the volume of dough in the measuring cylinder from the water bath at a temperature of 20 degrees Celsius at the end of the investigation. So the dough has returned to about 80. And how much was the dough in the beginning? Was placed at the bottom of a 200 cm cube, right? Now it says here, is to estimate the volumes of the dough at the end of the investigation in the other two measuring cylinders from water baths at a temperature of 40 degrees and 80 degrees. Well, at 40, it could be anything between 81 and 200, and in 80 degrees Celsius, anything between 50 and 79. To understand the question. Now, everybody has to be clear when they read the question, they must make a measure. In the second investigation, the student prepared three samples. Each sample of dough had a volume of 50 cm cube and contained all this stuff. Now, at 20, the dough rose up to 80 cm cube. So, at 40, it will be anything between 81 and 200. It will go a little more than 80. So, they've allowed anything between 81. I would have said 90, or I would have said 100. I would have said much more than that. And then it says at 80 degrees Celsius. Now the initial volume was 50, that we've just seen. The initial volume was 50 cm cube. Now if at 80 we think it has been denatured, so maybe it's just gone up by 5 or 10, maybe not, maybe just between 50, remain 50, or maybe 52 you can say. But 80 degrees Celsius should have denatured the enzyme, so it shouldn't have worked and the glucose should not have been available. But then yeast has its own glucose as well. So maybe it inspired that, and it was so any figure between 50 and 79. Why 79? Because at 20 it was 80. So it had to be something less than that. So this is why the figure for the 80 degrees Celsius was anything between 50 and 79. The reason you have to understand. Now coming to part 3, explain the results of this investigation. Well, that's very easy. What was being produced? Carbon dioxide was being produced. That's why the volume started to rise. And why? Because the enzymes were working. And 20 degrees Celsius is less kinetic energy, so fewer collisions. 40 degrees more kinetic energy, more collisions. Maybe it's the optimum temperature. At 80 degrees Celsius, it was denatured. Active site changes shape. Substrate no longer complementary. So all you can say the yeast is killed. So all that you have to give is an explanation of the results that we had. Why was the volume less or more, or why did you say something about the volume increasing? So this is for 3 marks. So the 3 marks were very easy to get if you had given me the basic points. The answer to that was simple. Explain the results. Carbon dioxide produced, and these carbon dioxide is produced as a result of enzyme action, and these enzymes are affected by temperature. So at 20 degrees, less kinetic energy, so less collisions. 40 degrees, more kinetic energy, more collisions, maybe it's the optimum. 80 degrees Celsius, the enzymes denatured, active shape of the enzyme changes shape, and it's no longer complementary, or you could have said the yeast is killed. So please understand why we are talking about this temperature. Unless you specify and tell me that enzyme is working, that's why temperature is having an effect on those reactions. Now, coming to question number 4, there are many different types of amino acid. One of these is phenylalanine. The diagram shows the structure of a molecule of phenylalanine. Phenylalanine is metabolized by an enzyme with the name PAH. Name the human organ that metabolizes amino acids such as phenylalanine. That's the liver. All amino acids, everything goes to the liver, and then, of course, it is either used up or whatever, or deaminated or whatever. Explain how the shape of a pH enzyme molecule is important to enable phenylalanine to be metabolized. Why enzymes? So you would just start off the enzyme story. So it's active site of the enzyme. So the active site of the enzyme is complementary or is specific, or you can say is lock and key. Then binds to or combines and then forms the enzyme substrate complex. So that was 3 marks. So how the shape of the pH enzyme? So you just have to give me all that. So active site complementary binds to substrate, forms enzyme substrate complex. So you had 4 points, but you could have given me any 3. Now it says a rare gene mutation reduces the activity of the pH enzyme. The mutation results in an increased concentration of phenylalanine in the blood. This condition may cause damage to the brain. Describe what is meant by the term gene mutation. It's a change or a modification or altering the gene structure or the DNA structure or the base sequence or the nucleotide sequence. So that is going to be a mutation. Mutation is actually change in the DNA. Change, for 2 marks, mutation is a spontaneous change in the gene structure, which of course gene means what? Genes, chromosomes are made up of DNA, and gene is a portion of the DNA which codes for a certain protein. So the change is in the gene structure or the DNA structure or the base sequence or the nucleotide sequence. So there are only two mark scheme points. Then let's explain how a person with the condition should change their diet to reduce the risk of damage to the brain. Eat less protein. Eat less protein, specific protein which has phenylalanine in it. Proteins are digested to and broken down to amino acids. So basically, you've got to just decrease the protein intake. So eat less protein, or you can find out the foods which are high in phenylalanine. So you have less of these, mostly it's dairy products, less protein containing food. And the reason is proteins are digested and broken down to amino acids. So we don't want that to be happening because it's phenylalanine is the one which is the culprit. So reduce the risk of damage to. Okay, now we come to the part 3. The gene mutation produces a recessive allele in a person with heterozygous genotype. What is heterozygous? Big H, small h. Is described as a carrier. A carrier will not show symptoms of the condition. Complete the genetic diagram. We said the gene mutation produces a recessive allele H. So that's given in the question. Complete the genetic diagram to show the probability of the two carriers reproducing to have offspring with the condition. So that means it's going to be capital H, small h, capital H, small h. So this will be capital H, capital H. This will be capital H, small h. This will be capital H, small h. And then this will be this one and this one. So it will be small h, small h. So now the gametes were of course H and small h here because they're both carriers. Offsprings will be these four. So probability of offspring with the condition, well, there's only 25% probability. You could have said 25% if you can said something else. You could have said 1 over 4, or you could say 1 in 4, or you could have said 1 ratio 3, or you could have said 0.25. So the probability is 0.25 of having. So basically, this is the one This is the one that's going to be the sufferer because it says the gene mutation produces a recessive allele. A person with a heterozygous is described as a carrier and does not show symptoms of the condition. So the people who are sufferers will be homozygous recessive people, which don't have the capital H but has the small letter h. Now we come to the last question. Question 5. Biological processes in plants are affected by environmental factors. The graph E shows the effect of changing one environmental factor on a biological process. The labels on the axis for each graph have been replaced with the letters P, Q, R, and S. Identify the correct axis label for each P, Q, R, and S. Select from the following possible labels: temperatures, carbon dioxide concentration, rate of transpiration, light intensity, rate of photosynthesis, and air humidity. Now for P, Q, R, and S. So P could be what? What we have to understand is they've given us the choices. So what is P? P has to be the rate of photosynthesis. And what is Q? Q is temperature. So P is the rate of photosynthesis. This was a typical temperature graph. So on the x-axis we'll have temperature and here we'll have rate of photosynthesis. Very typical temperature graph. So Q will be temperature and R and S. R will be rate of transpiration and this will be air humidity. As the air humidity is increasing, the rate of transpiration is decreasing. So P rate of photosynthesis, Q temperature, R rate of transpiration, and S is air humidity. Then we come to the last part. Describe the process of transpiration in the leaf of a plant. Evaporation from the surface of the cell wall of mesophyll cells, water vapor, is present in the air spaces, then diffusion of these water vapors down the concentration gradients and through the stomata. So basically the three marks were basically you have to start off with first the water, the film of water around the mesophyll cells first evaporating. So the point was evaporation from the surface of the mesophyll cells, and then the water vapors in the air spaces, and then the diffusion, and then water through the stomata. So evaporation, cell wall of mesophyll cells, water vapor, air space, diffusion of water vapor, and through the stomata. Now that completes this section A, and I hope this has become easier for you, as some of you mentioned that this was a little difficult for some of you to handle. But I hope it's easier. Please revise the chapter which you're finding it difficult, and do leave me the comments of how you would like me to improve any of this paper solving technique in any other way. Thank you very much.